So good morning to everyone. And first of all, I would like to thank Anne Walker for the nice introduction and also for giving us the chance to present what are really preliminary data on this TDARC gene. And what I decided to do today is just to give you an idea of which one is the main topic of our laboratory, that is Rett syndrome. So I'm going to do a brief introduction on Rett syndrome. And then what I would like to do is to instead recap with the data that we have already published and that I think constituted the rationale for having decided to apply to this call for grants on a TDARC gene. And then I will introduce the TDARC gene we got involved in that is named HPCAL4, and I will therefore show you what we collected so far in the last 6-7 months, because the grant started in July, but August is a sort of vacation month. So it's really 6 months probably that we are consistently working on that. So as most of you might know, Rett syndrome is a rare genetic disorder that affects mainly females with an incidence of 1 out of 10,000 live birth females. And although it's rare, because of its incidence, this is considered the main cause of severe intellectual disability in girls worldwide. The disease is characterized by what is generally called an early postnatal phase that appears symptom-free, but is not really symptom-free. And soon after this period, that generally lasts 6 to 18 months, the girls go through what is called a regression phase. And the regression phase is one of the hallmarks of the disorder. In the classical form of Rett syndrome, the regression is generally characterized by a short period that can last only a few weeks, in which girls start to show autistic features. They are not interested in the world outside anymore. They might be sort of continuously crying. They might have some sort of violent attitude. And in this brief period, they lose most, if not all, the skills that they previously acquired, meaning that if they were able to speak, they usually don't speak anymore, or if they were even able to say lala or something like that, they lose this capacity. They lose the capacity to use their hands, that instead it is substituted by a continuous stereotype hand movement that can be such as mousing, clapping, and washing, or something similar. And in this period, they also lose the capacity in general to move properly. After this regression phase, the disease stabilizes, and generally RTT patients arrive to adulthood. So the average lifespan is nowadays of 50 to 55 years old. But along the disease progression, a lot of symptoms gets in, and you can read that the main 1 year, so they suffer of a very severe intellectual disability, loss of expressive language, loss incapacity to use their hands, as I told you, stereotype and movement, apraxia, breathing abnormalities, really severe breathing abnormalities. They go that occurs mainly during the day and is leading to long apneas followed by hyperventilation. Most of them suffer of epilepsy, and these are generally drug resistance. They suffer of hypotonia, scoliosis, constipation, bone fragility, and these are just some of the symptoms. So you can really understand that the quality of these girls and their family is really poor. So it was back in 1999 that Uda Zogby discovered that the main cause of Rett syndrome stays on an X-linked gene that was already known and called MSCP2. As most of you might, since then we have proved that MSCP2 is in fact mutated in 95% or even more of classical Rett patients. And if you consider that we are sequencing only the coding portion together with the intron-exon junction, you can understand that this is probably a monogenic disorder, because we're not sequencing the introns, the promoter, and the long, highly conserved UTR, 3' UTR that MCP2 has, that is AKB of an UTR. And that, so what I was telling you is that since the discovery, MSCP2 was already known, and it is now very much characterized, and we generally can synthesize the activity of the protein by saying that this is an epigenetic transcriptional repressor that is able to bind to methylated DNA because it has this N-terminal methyl binding domain, meaning a domain of the protein that can recognize either CG or CH methylated dinucleotides and binding to them without almost any sequence specificity. Upon binding to methylated DNA, the protein is able to repress transcription, and it does this mainly, but not exclusively, through the presence of a transcriptional repression domain that here you can see in green, that is able to recruit on methylated DNA several chromatin remodeling complexes, and among these, the most important one are the ones that are able to recruit on methylated chromatin, or better, on chromatin as assembled on methylated DNA histone deacetylase activity. Further, the protein has a C-terminal domain that is less characterized, but since it can have even missense mutation in patients, this is clearly involved in the importantly also, and it seems to be involved in regulating the capacity of the protein to bind to chromatin and to DNA. The protein is ubiquitously expressed, but is really highly abundant in the brain. And in fact, it was the laboratory of Adrenberg that proved that in mature neuron, there is one molecule of MSCP2 every 2 nucleosomes. And because of that, and because of some biochemical studies and genetic studies, what is suggested is that the protein is in mature neuron will have a sort of architectural function, will substitute itself to histone H1, and therefore leading to a specialized chromatin structure. So all in all, what we generally believe is that MSCP2 is, since our genome and in particular neurons are highly methylated and globally methylated, and the protein is so abundant in mature neuron, the protein is ubiquitously bound or globally bound on our chromatin in neurons, and therefore makes a sort of difficult or to identify bona fide target of the protein, because it probably has global functions. So although I told you that this gene or protein is involved in Rett syndrome, we are now aware that the protein is also involved in several other neuropsychiatric diseases. And in these slides, the point I want to make is that we might have a loss of function mutation. These are in generally mutations that have a sporadic occurrence, so they generally occurs in the sperm, and they can lead to different kind of neuropsychiatric diseases. So we might have, as I told you, classical Rett syndrome or a variant form of Rett syndrome where the females are heterozygous for the mutation, or we might have males that are, of course, hemizygous because it is on the X chromosome, and therefore this male, if they carry a mutation that generally leads to Rett syndrome in women, they suffer from a very severe neonatal encephalopathy. On the contrary, they might have Rett syndrome if either they have a severe mutation, but this is in a Klinefelter condition, or they might have a very mild mutation that maybe in females don't lead to anything really, not a real serious issue in terms of neuropsychiatric disorders. On the same side, we might have that MSCP2 mutation, loss of function mutation, might cause in both gender other diseases such as autism, Angelman-like syndrome, schizophrenia, and learning disability. On the contrary, what we might have is that some patients suffer from what is now recognized as a distinct disorder, and is called the MSCP2 duplication syndrome. In this case, we're going to have mainly boys that are carrying 2 or 3 copies of the wild-type MSCP2 gene, and they are suffering of a very severe neurological condition where the main features are severe epilepsy, severe cognitive or intellectual disabilities, and also recurrent infections. In this case, the duplication is inherited by the mother, that is a carrier, and the mother is characterized by the fact, in general, to have a skewed X chromosome inactivation. So in general, what is the chromosome that gets inactivated is the mutant one. And in spite of that, what is clear is that generally these women have some, again, neuropsychiatric conditions. So they might have some sort of bipolar disorder, some form of depression or schizophrenia. So all in all, we believe that almost one human being out of 6,000 suffers from some condition that are related to MCP2 mutation, highlighting the importance in better understanding what is this, how is this protein functioning, and how can we manage or treat the disorder that are linked to this gene. So, as you can imagine, mouse models of the disorders have been and still are instrumental, both for basic studies and for preclinical studies. In general, several mice have been generated. So we have null lines, we have knocking, conditional, and so on. But the ones that are still mainly used are the null lines that have been generated both at the Jenisch laboratory and Adrian Bird laboratory. Generally, what most of the people still study or use for the study are the hemizygous male null mice. And the reason of that is because, as you will see, the disease is more consistent and is also, the onset is quite early, so it is easier to study in this animal, although we are aware the disease is typical of an heterozygous condition. Furthermore, since the males are sterile, we need the females for maintaining the colony. So anyhow, to summarize the features of these animals, we can say that the MSCP2 null male mice are characterized by being born in an apparent normal way. So they seem to do fine for almost 30 days when they go through again a regression phase and they start to manifest their symptoms. And if we look at the symptoms, we can say that they generally suffer from clasping, that is a clear indication in the erection of a neurological condition, locomotor deficit, hypotonia, ataxia, seizure, breathing abnormalities, the deficit in memory and learning, and so on. So they really well recapitulate the human disorder. And the lifespan, however, of this animal is very short, as it is the case for most of the cases for the MSCP2 mutant boys. So they generally lifespan is in between 80 to maximum, I would say, 100 days. And concerning the females, the heterozygous females do show the same symptoms, but the onset is later. So you might not see anything for the first 3 to 4 months. And they are milder. And because probably of X chromosome inactivation, and also all the symptoms are more more heterogeneous. So we might have more different entity of the symptoms depending on the animals. So making the studies, of course, not only longer, but more difficult because everything is milder and it requires more animals to get significant results. But we are aware that this is the best model to study the disease, whereas the null animals are pretty good for understanding the function of MSCP2. The study that maybe I want to mention 3 more studies quickly. So conditional mice have been used to prove that the disorder is mainly a disorder of the central nervous system. Neurons are the cells that are mainly affecting the different symptoms, and depending on the subset of neurons, and if you remove MSCP2, you will find out a different subset of symptoms. But anyway, even astrocytes participate to the disorder. And the other important information that was obtained from conditional mice was obtained again from the laboratory of Adrian Bird. And what he did is to generate a mouse line that was floxed. So it has a stop codon in the MSCP2 gene, but close by to the stop codon, there was some flox sequences. So what he did was to have animals that were born affected by Rett syndrome, and then to induce the removal of the stop codon and therefore reactivating at physiological levels the MSCP2 gene at different ages along the disease progression. And what he clearly proved, both in females and in males, is that at least in mice, the disease is completely reversible. So the animals could live more than 1 year, and the mice that were reversed, they didn't show almost any more phenotype. They were showing some, still maintaining some tremors, but most of the other symptoms were gone. So suggesting that there is no neurodegeneration, so this is not a neurodegenerative disease, and that, of course, there is some hope for treatment. And to sum up very briefly, what are the main features of the brain of the RTT mice and also of the human patients? I can just say that everything is in place, and of course this is the case since the disorder is reversible. But what it has been clearly proved is that both in human being and in the mice animals, there is a clear volume reduction of the brain. This we can measure this quite early, even in the mice earlier than when the symptoms get really overt. And this reduction is affecting any brain region, but with different entities, depending, of course, on the specific brain area. And what is the reason of this volume reduction? We believe that is mainly given, is not given by the loss of neurons, because as I told you, this is not a neurodegenerative disorder. So the cell numbers, the number of neurons is maintained, but they are characterized by having a smaller soma, and they are characterized by having reduced dendritic carbonization. So the maturation of this neuron is impaired, or the maintenance of their maturation is impaired. And what it has also been proved is that there are a clear defect in spines, so there are fewer and immature spines, and in accordance with that, there are problems in spine plasticity, and we have aberrant LTP and so on. And also we are going to see that there is a disruption in the excitatory and inhibitory balance, but what is important also to highlight that this might have a different, the disruption might go to different direction depending on the brain region that we are analyzing. So for example, the cortex is less excited, whereas the hippocampus is more excited. And this makes sense because we are all aware that MECP2 is a global regulator of gene expression, but it affects epigenetics, or it reads epigenetics. And therefore we are all aware that epigenetics changes depending on the specific neuronal type, brain area, and of course, depending also on environment. So let me just finish this introduction by saying that no cures are still available. There are 2 clinical trials right now going on for gene therapy. They just started, that they are enrolling the patients, and instead that the only molecule that passed the stage 3 of a clinical trial is this lavender, that is trofinetide, that is a sort of IGF-1. So it mimics the IGF-1, the neurotrophin. So we might end up with the next year having the first molecule for a neurodevelopmental disorder. So having said that, let me say why we started to get involved with this HPCAL gene that, as we said, is a TDAR gene. Everything started several years ago when Francesco Bedoni was in my lab, and he was a postdoc that was very much interested in cortical development. And Francesco is right now instead a postdoc in Cardiff. And so since he was interested in cortical development, he started to be fascinated at the idea to understand if MECP2 was playing any role during cortical development. And the reason why I did this study is because since then, all the studies were mainly focusing on the either, I would say, adult phase, or when the regression phase gets in both in mice and again in human beings. So as you can see from here, most of the study were looking at postnatal neurological functions in the Rett brain. So instead, he decided to see if what was going on during embryonic development. The first thing he had to do is to make sure that MECP2 was expressed in the embryonic cortices, and so in the MECP2 defective embryonic cortices. It was expressed in the wild type embryonic cortices. So what he did is to use different immunohistochemistry approaches, and he was able in this way to prove that in accordance with literature, MECP2 is more expressed in the more mature cortex, but actually the protein is expressed at any stage, and it is even expressed by neuroprogenitors. So having proved this, what he decided to do is to look at the transcriptional profile to compare compare the transcriptional profile or wild type and MACP2 null, so we're talking about a male embryonic cortices, and therefore he performed the analysis at E15. So to cut the story short, what he found out using a GSEA analysis of the different found a high number of differentially expressed gene in the null cortex, and these genes are mainly downregulated, and this is in accordance with several transcriptional profile studies that have been performed at any age in the Rett brain. And also the entity of the fold change of the dysregulation is very little. So many genes, but very little deregulated. And this again is in accordance with the idea that this is a sort of a dimmer of transcription. MCP2 is really finely regulating transcription. So anyhow, by using GSEA, he was able to prove that if we look at, we compare the null pathways, we compare the null and the wild type cortex, we can find that in the null cortex, there is an enrichment in genes that belong to the pathway that we can classify as important or typical of a proliferative identity. And on the contrary, in the null cortex, there is only in the null developing cortex, there is not enriched, or is much less enriched with respect to the wild type. There are much less enriched genes that are involved in post-mitotic identity and in neuronal maturation. So in other words, what he found out is that there was a delay in, there was a profile that was suggesting a delay in neuronal maturation and cell commitment. So he also discovered that in accordance with that, there were several genes that are involved in neuronal activity that were defective in their transcription. And this was real during cortical embryogenesis or in the perinatal age, as you can see here from P8, but was even real in cultured neuron. So by comparing again wild type and null samples, he found out that the null samples are defective in several genes that are important for the capacity of the neurons to get stimulated or to respond to stimuli, such as AMPA receptor and MDA receptor, calcium channel, chlorine channel, potassium channel, sodium channels, and so on. So all in all, this led Francesco to hypothesize, and at that time Linda Scaramuzza joined the laboratory, and Linda today nowadays is a postdoc at INGM in Milano with Professor Elena Cattaneo. And so they started to think that probably this impairment in gene transcription might have led to neurons that were impaired in responding to stimuli. So, and in fact, they proved this simply by calcium imaging. So if you stimulate wild type or null neurons with glutamate and you record calcium imaging, what you can find out is that the null neurons that here are in red are characterized by having a decreased response to the stimulus at any age of an analysis. So since the early development in culture, they were really demonstrating an impaired capacity to respond to stimuli. And so this finding motivated Linda to perform a PhD in our laboratory in assessing the relevance of this impairment for the pathology of Rett syndrome. And all of this was motivated by this review by Spitzer, in which Spitzer started to claims that mainly during early development, but actually during all our brain life, there is a feed-forward circle between transcription, neuronal activity, and morphological complexity in neurons. And in particular, its idea is that a proper transcriptional profile is essential to obtain a proper capacity of the neurons to respond to stimuli, and this capacity to respond to stimuli affects neuronal morphology that on its side will again affect transcription, and so on and so forth. So what Linda started to think, okay, here we are facing instead with a disorder in which we are lacking, or better, we have a master gene of transcription that is not functioning in a proper way. So transcription will be impaired. As a consequence, we will have a minor capacity to respond to stimuli. So we are going to have an affected neuronal activity, and this will affect the neuronal morphology. But if the neuronal morphology and the neuronal activity are not working properly, transcription will again be more affected. And so this is becoming a sort of vicious circle. And so she decided if this hypothesis was correct, and therefore in the Rett syndrome, because we're going to have transcriptional defects, all the neuronal maturation, the capacity to refine circuits and to maintain mature circuits is affected, she thought, well, now we can think of a slightly stimulating the RTT neurons, so meaning just giving them some low level, but some stimuli in order to increase a little bit the tone of these cells and try to figure out if this could improve the morphology, the activity, and the network of the cells. So she performed this at the beginning in vitro. I'm not going to show the data. And by doing this in vitro, she was able to prove that the hypothesis was correct. So she could rescue dendritic arborization, neuronal morphology, the capacity to respond to stimuli by calcium imaging, and she could even ameliorate significantly the transcription of several genes. So because, okay, I forgot to say that she obtained this with two different approaches, either by treating neurons with a tiny amount of KCl. So she just provided 4 millimolar of KCl to the culture, in addition to the one that might be present in the medium. And or the other thing, she instead provided ampakine. And the ampakine are drugs that are able, by binding to glutamate receptor, are able to slow down the inactivation of the receptor. So meaning that we are just maintaining the receptor open longer, but we are not activating receptors that are not stimulated. So having obtained all these positive results, what she decided to do then was to try to assess if this could be a sort of effective also in vivo. And we decided to look at the treatment in an early time window. We are aware that that treatment was not a translational treatment, because we gave the ampakine from P3 to P9, so really in the perinatal age that does not correspond to a period in which the disorder is already diagnosed. But the point was only to check if by stimulating the activity we could have a positive effect. So what she did, again, she treated the animal for 6 days, either with the vehicle or with the drug, and then we assessed the general well-being of the animals, the life expectancy, and again, these are null male animals or wild type, treated or not with the placebo or the ampakine, and the analysis have been then performed in blind by an operator. And so, but the behavioral assessment was performed on average at P45, so meaning 35 days or 40 days later than the last day of treatment. And to our surprise, we could find out that the treatment was quite effective. So we could find out that there was a significant elongation of the life expectancy of the animals. And the animals, this is a scoring system that we generally use in when we study Rett mice, and where we generally assess what is the general well-being of the animals, tremors, locomotor functions, and clasping, and we give scores, and the highest is the score, the worst is the phenotypes of the animals. So you can clearly see that, of course, the wild type animals don't have any symptoms, so they don't have scores. And on the contrary, the null animals that are treated with the vehicle are accumulating always more severe scores. And on the contrary, the disease progression is slowed down in accordance with the life expectancy in the treated animals. So in accordance with this, what also Linda and Giuseppina De Rocco, that is still working in the laboratory and is performing a PhD study in which she want to try to find out a better treatment with ampakines, but anyway, this is not the topic of today. But what they did is to look if the treatment with the ampakine could ameliorate the mobility of the animals and the capacity to and the coordination of the animals. So they use for this the Rotarod assay, and what you can see is that if we compare the null animal with respect to the wild type, and this is the null animal treated with the vehicle, we have a significant defect that is not present anymore in animals that were treated with the ampakine, and the null animals that were treated with the ampakine, suggesting that there was an amelioration even almost 40 days after the treatment. On the same side, we obtained the same result in cognitive function. So we performed a novel object recognition test. The paradigm was used was testing what we call the long memory. And again, we could find an almost significant defect in the null animals that was instead not present anymore, and is clear the amelioration in the treated animals. So all in all, leads us eventually to why we decided to study on this TDARC gene. I don't have to introduce the call that gave us the idea to start to study this TDARC gene, but again, this was a call funded by Fondazione Cariplo and Fondazione Teleton, and as Anna told you, the bottom line of this call is that they want really to try to make a researcher to focus more if a TDARC gene or any TDARC gene might be involved in our of the rare diseases that we are all studying. So we found this really fascinating, and therefore we decided to start to look if by any chance there was some TDARC gene that might be involved in the disorder and that we didn't notice. And so what we decided to do at first was to look at our data, RNA-seq data that have been generated along the years in several studies. They are mainly preclinical studies or in order to start the drugs. And these have been performed in cerebellum, cerebral cortex, and so on. So, and what we found out is that the TDARC gene HPCAL4 that I will introduce in a second was consistently downregulated in the knockout again, so it's a male null brain. And this was performed, was seen in the adult brain, so at P60, so meaning that this is a symptomatic mouse, quite severe mouse already, and was present in the cerebellum, in the cerebral cortex, in the hippocampus, but was even present in neurons that were cultured in vitro at D14, and it is, and we also perform RNA-seq data that we will never publish because I don't think the synaptosome that were purified were really, really clean, I would say, but we could find out that HPCAL4 was looking like highly enriched mRNA in synaptosome, and it was downregulated in the null synaptosome. And I can tell you that the presence of HPCAL4 in the mRNA, the mRNA for HPCAL4 in the synaptic compartment has been confirmed by a recent publication in which they found out that it is highly enriched in the postsynaptic compartment. So all this data were really interesting to us because, as I told you, it is really, since MSCP2 is a fine modulator of gene expression, it's generally difficult to find a consistent downregulated in different brain regions. But we wanted to make sure that the data were consistent with our other laboratories. And so we looked at on databases, and what we could find out is that, for example, it was already proved as downregulated in RNA-seq studies and even proteomic studies performed, obtained in mouse cortex of null adult animals. And as you can see from here, the defect at the protein level is bigger than the mRNA level, suggesting that probably there are also some post-transcriptional regulation. The defect is present in the mouse striatum, and importantly to us, because it had some translational value, it was found defective also in some cerebellum obtained from some patients that pass away. So all in all, this gave us the idea, okay, to try to check if HPCAL4 might, what is the function first of HPCAL4, and then if it might be important for Rett syndrome. So what is known about HPCAL4? So HPCAL4 is called a bilipin gene, and it belongs to the visinin-like subfamily of proteins. What are these proteins? These proteins are all calcium sensors, and they are all characterized by the fact that apart from having domains that are important to bind to calcium, they do have a link to their N-terminal region, a myristoyl group. When the cell is in a so-called basal condition, the hydrophobic region, there is an hydrophobic region that is maintained inside the protein, and this contains the myristoyl group. But when instead the calcium is able to activate the protein changes its own conformation. The myristoyl group can be exposed to the outside, and because it connects with either phospholipids or proteins that still remain to be discovered for most of these calcium sensors, it can associate with the membrane. And in fact, what we are known is that all these proteins have this, all these bilipin proteins are able to sense calcium. They might be sensitive to different amounts of calcium concentration, and it is nothing is known about HPCAL4 on these topics. And upon activation, they might translocate to different subcellular membrane compartments, so for example, the Golgi or the plasma membrane or whatever. So what in generally we believe is that this neuronal calcium member, that calcium sensors, sorry, there are 5 members of these families, and they can be considered as proteins important to transduce a signal and to therefore affect the neuronal signaling. It has been proved that some of them are involved in regulating and affecting the MAP kinase signaling, some the cyclic nucleotide signaling, and others might be involved in affecting the trafficking of different receptor and ion channels. So concerning HPCAL4, very little is known. It has been hypothesized and somehow proved that the protein is involved in binding to the presynaptic Cav2.1 channel, where it probably inhibits, and by binding to it, it probably inhibits inactivation and therefore enhance the facilitation of this channel. So considering all above, okay, we assume that maybe there was a connection between the incapacity of the Rett neurons to respond to the stimuli and the deficiency of this HPCAL4, and therefore HPCAL4 might participate to this minor capacity to respond to stimuli or to transduce its signal. And because of that, we proposed our grant that, as said, got funded. And the main idea of the whole grant is to try to understand if the alteration of HPCAL4 is expression can be causally linked to the neuronal deficit observed in RTT. And therefore, the idea is to try to understand where and when HPCAL4 is expressed in the wild-type brain and in the RTT brain, also defining, therefore, which region are defective in the expression of HPCAL4 at the protein level, mainly, and when this defect starts. And also what we wanted to do is to try to understand where is the protein localized in neurons that can be resting or stimulated, and then to address what are the function of HPCAL4 in wild-type neurons. So by silencing it, we could evaluate the different a wider series of electrophysiological measures in order to understand again if it is involved in synaptic transmission, plasticity, LTP, and so on, and also if it is involved in exo or endocytosis at presynaptic terminus. Eventually, what the idea was to identify partners in order to better understand its function, and then to go back to the Rett neurons and give back to the Rett neurons, or therefore to transduce in Rett neurons HPCAL4, increasing its level in order to address if we could restore typical RTT phenotypes that could have been triggered by the deficiency or triggered or might have been a contribution by the HPCAL deficiency. So what we have done so far we have analyzed HPCAL4 protein expression. We focused on protein because of this possible post-transcriptional regulation in wild type and RTT mice. So we focused at the beginning in wild type mice in order to understand when the protein is expressed and mainly where. So we started to look at ages, so immediately after birth, when synaptogenesis is starting at P7, when synaptogenesis is almost over at P20, then in adult brains. And that initially we focused on cerebral cortex and hippocampus that are generally considered the two main targets of Rett syndrome disease. And so as you can see here, from here, you can clearly see that there is an increase of HPCAL4 protein upon brain or maturation. And this start to peak really at the end, I would say, or almost the end of synaptogenesis. What is also interesting to find out that this is, however, is not true for any region. And so what we found out is that if we look at the cerebellum, we're going to see the same raise of HPCAL4 in the cerebellum, although the level are, well, we can go to this later, the same raise of HPCAL4 from P7 to P20, but then the level of the protein drops importantly. And this is probably the reason why HPCAL4 is described as not present in the cerebellum if you look at brain atlas in mice. So we then also decided to check also in wild type animals which are the brain regions that are mainly expressing at the different stages the protein. And we focused again on cortex, hippocampus, and cerebellum. You can clearly see how much less the protein there is in the cerebellum, but we included the brainstem and the hypothalamus that might be involved in the disease. And the result that we obtained is that clearly the cortex and hippocampus are mainly or more expressing HPCAL4 with respect to other tissue. And because of that, and because we generally culture either cortical or hippocampal neurons, we decided to focus on these two brain regions, but include also the cerebellum, at least for some studies, because I showed you that in patients it has been found a defective protein HPCAL4 level, so in Rett patients. So because of that, then we moved on and started to compare the level of HPCAL4 in the RTT brain. So here you're going to see the analysis performed at P7 and P20. These are 2 times in which the protein is the protein in which the disease does not show overt symptoms. So generally we call this the so-called asymptomatic period. And what it is clear is that both in the cortex and hippocampus, and P20 was also analyzed at P20, there is no defect in the HPCAL4 expression. On the contrary, the defect becomes overt at P40, so when the disorder is now, when now the symptoms are overt. And we could find a quite relevant decrease that is almost of 50% in, and sometimes even more in the cerebellum in all the tissues that we analyzed. And if we look instead when the animals are really in bad shape, and I want to mention the fact that there are a lot of compensatory mechanisms that are occurring at that ages, what we found out is that the defect was clearly still present in the hippocampus and the cerebellum, but it was not significant anymore, although there was a clearly decrease in the protein levels. So all in all, I would think that these results show that yes, the protein is downregulated in the brain, however, of the symptomatic mice. So suggesting that somehow maybe we can claim that at least at the protein level, this is not a primary target of MCP2, but is a secondary consequence of the deficiency of MCP2. But we have to see, because at the transcriptional level, this is not exactly the case. So then we decided to see, okay, we are aware that the females are the best models. So we wonder, can we reproduce some sort of similar phenotypes in the female animals? We are aware that females generally have a really milder phenotype, so it's difficult to detect them. But we selected for the time being, we're going to just one point that there was a quite symptomatic time points because it corresponds to P200. And at P200, we could find again a defective HPCAL4 expression in the cerebral cortex, but this was not present in cortex and hippocampus. So we're thinking now to go instead to anticipate the time of analysis to try to understand if what we found out in the male animals that at one point we do not detect anymore the symptom might be even true for the females. So having done this, what we started to instead study was we wanted to detect if we could find a defective protein expression even in cultural neurons, and also to start to address where is HPCAL4 localized in cultural neurons. So in order to respond to this, at first we decided to find out when the protein is clearly expressed. So again, we performed the Western blots, and what you can clearly see is that the level of HPCAL4 are increasing with the timing of culturing of our hippocampal neurons. So again, suggesting that in accordance with the brain, the protein seems to increase its levels with neuronal maturation. And therefore, what we decided to do is to evaluate at the DIV19, which is the level, if there is any, and which kind of defect is present at protein level. And what you can clearly see is that at that age, again, we have a clear defect in cultural neurons. So the question then we wonder, you can clearly see that here the defect is really important, whereas in tissue it was less important. So we started to wonder, so is this caused by the fact that, for example, in tissue there are astrocytes or glia, and maybe other cell types might not be affected by this defect. So what we decided to do is to try to figure out if we could see in cultured astrocytes the presence of HPCAL4. These, however, are since they are cultured, they are immature astrocytes, but we have not been able to detect the expression of the protein in cultured astrocytes. So it seems to be in accordance with most of the bilirubin protein that this is a neuronal, is a protein that is mainly expressed in neurons. So we also wanted to address if we could find its expression in other tissues. So according to other bilirubins, we went to look at the spleen, pancreas, kidneys, and lungs. So we didn't detect any protein by Western blot in spleen and pancreas, very little expression of the protein in the kidney, and not sufficient to quantify it, whereas in the lung the expression is a little bit higher, and this permitted to compare the level of expression. And what is clear is that there is no defect. So we're sort of inclined to saying that the protein is mainly a neuronal protein, and anyway the deficiency in Rett syndrome is mainly associated with neurons. So having said that, we decided to look at the expression of the protein, so where it is localized, difficult to say. We are not able by immunofluorescence to detect it. So what we decided to do is to generate a construct in order to follow it. So we just generated a wild type construct in which HPCAL4 is linked to a MYC tag, and we also generated a G2A mutant that cannot be myristoylated. So in order to make sure that the protein was behaving in the proper way, we fractionated ECK cells that were transfected with the wild type or the mutant, and you can see that in accordance with the expectancy, the HPCAL4 wild type protein goes into the membrane. So this is a cytoplasmic fraction, this is a membrane fraction, are not stimulated or stimulated with calcium, and you can see that there is an enrichment in the membrane fraction, whereas on the contrary, the G2A mutant is unable to move into the membrane as expected. It was also interesting to find out that the HPCAL4 mutant protein was somehow more expressed in ECK cells. We don't know what is the meaning of that, but it's very much consistent. And it's something that it is worth probably studying because of the studies on hippocampal neurons that I'm going to show you right now. So having the right construct, we decided to transfect the wild type HPCAL4 protein or the mutant one in hippocampal neurons. And these are at D14. They have been stained either for MYC or they've been stained with MAP2 to follow the dendrites. And what you can clearly see from these images is that the protein is practically all over in the dendrites. It's very clear that the soma is almost empty. So in accordance with the idea that it's mainly localized towards the membrane, and you can clearly see that that it goes into spines. So we tried also to obtain data with the G2A mutants, but although in the vicinity of these neurons, there were good-looking neurons, so far, but these are preliminary data again, we have not been able to find well-arborized neurons. So this might mean that the mutant is somehow toxic for the hippocampal neurons. And so this is why I'm interested in trying also to address if it is really much more expressed. But anyway, we also looked at the signal intensity in order to understand if the protein was indeed localized at the membrane level. And it appears that this is the case for the wild-type protein that is enriched at the membrane. This is not the case for the mutant, as I expected. So, and let me also say that we didn't find any localization inside the soma, so we don't think that this is a protein that goes at the level of the Golgi. So we then stimulated the neurons, with the wild type protein, and start to show this sort of foci of MYC, so we'll start to see oligomerization probably of HP-Cal, as it has been proved already for hypocalcin, that is one of the 5 bilipin genes that belong to the same family. So it looks like that it gets to aggregate or to oligomerize upon stimulation. And again, if you look at what is going on in dendrites, although we are aware that these neurons are not feeling well, so I will not take it too much in consideration, but we couldn't see this agglomeration or this sort of oligomerization with the G2A mutant that instead remains soluble. And again, the localization at the level of the membrane is maintained. And eventually, and I'm done, what we're doing about molecular partners, we have no data about that. I want to just tell you how we are approaching this. We are approaching this using either co-IP, simply by immunoprecipitating HP-Cal4 from brain, and then looking wild-type brain and looking for interactors. So we think the protocol is set, because you can see here that we have immunoprecipitated HP-Cal4, whereas the negative control, where the primary antibody was omitted, is quite clean. So now we can scale up this protocol and check for interactor by mass spec. So this is a good test because it permits us to go to look at brain, but we also wanted to use proximal labeling in order to find out interactors where, so as you might know, what happens is that you fuse your target gene, such as HP-Cal4, to a turbo or a mini turbo in our case, and this is a protein that is able to activate, substantially what it does is to generate a biotinylated AMP, and this is a reactive intermediate that can biotinylate anything that is really close by to your target genes. So this is a trick that has already been used to identify partners in different systems, as well as in different compartments of the cells. So we have fused HP-Cal4 to a mini turbo. Of course, the mini turbo has to stay at the C-terminal portion. We proved that the fusion is not affecting the capacity of HP-Cal4 to get into the membrane. And we have just performed the first experiment to see if it is working. So here our cells, X cells, are transfected with the mini turbo, and here they are transfected with the HP-Cal4 mini turbo. Here you can see that the protein biotinylates itself, and this is correct because it is HP-Cal4 can be biotinylated by mini turbo. It is very close by, but we could already see that there are some proteins that are probably not present in the control. And so again, we have to scale up this and perform mass spec, but we are hoping to do this in neuronal cell lines. So let me finish just by thanking the person that got involved mainly in, that are involved right now in this project. And in particular is Stefano Pezzini, that is a postdoc that joined our laboratory thanks to this grant and is working on that. He does this in collaboration with a PhD student that just joined the laboratory from Lebanon. And previous studies with some Western blot were performed by Alessandro Arcari, that is a fellow fellow in our laboratory, but is now focusing on a different project. The imaging studies have been performed, oh, I forgot the name, I'm sorry, but no, I have both by Lucrezia Scandella, a fellow that is under the supervision of a great professor that is excellent in imaging, that is Maura Francolini. And on the contrary, the experiments of electrophysiology and also the transfection are performed in collaboration with Davide Pozzi and a postdoc at Humanitas that is Edoardo Fraviga. And these are the funding agencies, so the joint funding from Fondazione Cariplo and Teleton, and an association of parents that is always funding our laboratory. And I thank you for your attention.
Thank you. Thank you very much. I would encourage, if anyone has questions, to please put them in the Q&A, and we'll be able to address them there. Let me see here. So let me actually just ask, in the transition of the development of this disease that you have in humans, in the diagnosis, are there any kind of assessments or diagnosis elements that's done with genetic assessments of these expression of these? Is that ever a potential for these dark targets, so that early on you could see that this is a person who is showing maybe not all the neurological symptoms, you know, but genetically have?
No, yeah, I got so let's say that for the time being, of course, being a TDARC gene, this is not included in any genetic screening, and I didn't find anything that might suggest its involvement. But for example, there is another neurosensor that is linked with dystonia. And these girls anyway are very much suffering from ataxia. So I don't know, not now, of course, this is not the moment for including this in genetic tests, because we need to gather some. My idea is that it might be a modifier of, I don't think it will cause Rett syndrome, but it might modify the phenotype. So I didn't have the time to say that patients might have different severity of some disorders. Sometimes they share the same mutation, but one patient is very severe and the other one is not, although they seems to have the same chromosome, X inactivation chromosome pattern. So this idea is that they might have modifier genes that are protecting or exacerbating their phenotypes. And in fact, it is known that there are carriers, mothers that are carriers of the disorder. They don't show any phenotype, although the sons or daughters that are going to have are severely affected. So saying that, I think studying HPCAL4 might be relevant to understand if it could be a modifier of this neurodevelopmental disorder or other neurodevelopmental disorder.
Gotcha. Thank you. Got one comment into the question answering is from Jeremy Yang. Bravo, excellent and inspiring work. I'm going to do one more call to see if we have any more further questions. I also would say that, you know, as these seminars are to inspire collaborations and interactions, I'm sure if you have any questions further that you come up with, kitty cat, yay!
Sorry about that.
If you have any more further questions, I'm sure Dr. Landsberger is willing to answer them via email as well. So feel free to email.
It would be my pleasure.
Great. Thank you once again, and have a lovely evening there in Italy. Thank you.
Thank you, and have a good day, you.
Thank you.
Bye.
Bye-bye. Bye.